Hello everyone, welcome back. Welcome back to day 2 of topic 10. In today's lecture, what we have left to talk about is meiosis and then bacterial and archaeal cell division. And again, we're working on answering our overarching question of how do we make more cells? So we're going to pick up with meiosis, so part 4. And before we get into an overview and before we get into a lot of drawing, let's first talk about why do humans even need to undergo meiosis? So why do our cells need meiosis? So that we can do sexual reproduction. So again, I encourage you to pause the video, and if I answer this question in a different way than you are thinking about it, then in the comments below, as always, share your thoughts and ideas. So if we think about this, if we have 46 chromosomes, if we take 2 diploid cells and they each have 46 chromosomes and we smash those together, now we have a cell with 96 chromosomes. And so now instead of having 2 pairs of everything, we have 4 pairs. So instead of being diploid, we're tetraploid. Essentially what happens is we amplify exponentially every single round. But if we think back to humans, well, we have 46 chromosomes, and if we were to go on to have progeny, our progeny would also have 46 chromosomes. And the only way that is possible is to undergo meiosis, which goes from a diploid cell to haploid cells, and then allows us to take 2 haploid cells and to bring them together. So if we have an oocyte or an egg, and I always like to draw my eggs and sperm with a bow tie on them, but eggs have 23 chromosomes, so they are haploid, n equals 23. And they're going to be fertilized by sperm, which also have 23 chromosomes. And those gametes are then going to come together to fertilize the egg, and we get 2n equals 46. So the zygote then becomes diploid. So we go from 2 haploid cells to 1 diploid cell. And again, if we didn't undergo meiosis before we underwent sexual reproduction, then we would have progeny that would have double the chromosomes, and that would just amplify exponentially every single round. So let's start with a general overview of meiosis. And what we're going to do is remind ourselves of what happens in mitosis, and then we'll draw meiosis below. So here we go. So in mitosis, we start as diploid, and let's just continue on with 2 pairs, so 4 chromosomes. At the end of mitosis, we end up with 2 daughter cells, and those 2 daughter cells are both 2n, and they are identical. On the flip side, in meiosis, even though we start with a cell that is 2n, we end up with 4 daughter cells, and each of those daughter cells are n or haploid, and they have half the amount of DNA. And when we think about meiosis, we're not going to go into as much depth as mitosis. What we're going to do is we're going to talk about the key similarities and key differences when we compare mitosis and meiosis. And so what I will say is when we talk about meiosis, this is essentially 2 rounds of mitosis. So let's go ahead, and just like we did with mitosis with our blank page, we're going to draw the process of meiosis. But because we already know the steps of mitosis, so that mat that we talked about, we're going to condense pieces of it. And again, with the goal here is to focus on the main similarities and the main differences. So let's start with our blank page, and we're going to talk about an organism that has 2 pairs of chromosomes, so 2n equals 4. And what we're going to do is we're going to draw what this cell would look like at interphase and at the start of interphase. So at the beginning of interphase, if we have 2 pairs, we'll keep with our same color scheme that we had on day 1. So our orange and our pink. So we have an organism that is 2n equals 4. And we go through replication. So we're going to go through S, and we're going to go through G2, and we're going to draw the chromosomes already condensed just so that we can better see what's going on in this nucleus. So after those chromosomes have been copied, they have that X shape. So we started with 4 chromosomes and 4 chromatids, and now we have 4 chromosomes and 8 chromatids. Just to remind ourselves of some of that terminology, those pairs, we have 2 pairs, so we have homologous chromosomes. So recall from day 1 that when we talk about pairs, we also have to think about homologous chromosomes. So we have 2 pairs or 2 pairs of homologous chromosomes. And then we are going to go through meiosis 1. So interphase, whether it is for mitosis or meiosis, is the exact same. We go through and we replicate the DNA, and again, we'll have an entire topic dedicated to how DNA is replicated, and we're going to go through meiosis 1. Now, if you recall from mitosis, in mitosis, we separate the sister chromatids. In meiosis 1, however, we pair the homologous chromosomes and we then separate the homologous chromosomes. So one thing to keep in mind, the key for meiosis 1 is that sister chromatids do not separate yet. So at the end of meiosis 1, we will have 2 daughter cells, but because we separated the homologous chromosomes, those sister chromatids are still stuck together. So again, the key here is the sister chromatids do not separate. Only the homologous chromosomes are paired and then separated. So if we were to zoom in here, so we'll draw a little picture. And again, we've kind of taken the steps here of meiosis 1 and condensed them into one, but the same overall process happens that happens in mitosis. So here we have our duplicated chromosomes and we're ready for meiosis 1. So in meiosis 1, just like we get in mitosis, we get the breakdown of the nuclear envelope and we get the formation of the spindle fibers, and we get those homologous pairs that are going to, and we get those homologous chromosomes that are going to line up in the middle. The spindle fibers attach to the chromosomes. Those chromosomes are then pulled apart, and again, we are separating the homologous chromosomes, and we will go through telophase, so we will remake the nuclear membrane again, we'll go through cytokinesis, and our end result will be 2 daughter cells in which the homologous chromosomes or the chromosome pairs have been separated, but the sister chromatids are still connected. And now, at the end of meiosis 1, now we're ready for meiosis 2. And meiosis 2 is most similar to mitosis. And what I mean by that is our sister chromatids now separate. When the sister chromatids separate during this step, we actually do not do another round of replication. So we go from the beginning of interphase, so G1, go through SG2 where we have a replication. We then enter meiosis 1. At the end of meiosis 2, we have 2 daughter cells that have the homologous chromosomes that have been separated. So they still have the sister chromatids attached. And now at the end of meiosis 2, we will have 4 daughter cells and those 4 daughter cells will be haploid, and they will each have half of those chromosomes from meiosis 1. So we start with 2n equals 4, and we end with n equals 2. So again, if we zoom in and what happened in those steps, we had the PMAT that happened. So we had prophase prophase 2, where that nuclear membrane starts to fall apart. We start to make our spindle fibers. Those spindle fibers are going to then attach to our sister chromatids. So I'm going to draw just the pink cells just because I'm running out of space here. So in metaphase, we're going to have those pink chromosomes lined up in the middle. And then in anaphase, we're going to have those pink chromosomes, specifically the pink chromatids, the sister chromatids, right, that will be separated. And that, of course, will lead to having our 4 daughter cells. So in meiosis 1, we have prophase, metaphase, anaphase, telophase, and cytokinesis, and we call it prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. In meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. And again, here what I want you to focus on for meiosis is what goes in and what comes out, and knowing what is separated at each step. So in meiosis 1, the homologous chromosomes are separated. In meiosis 2, the sister chromatids are separated. So here is an image, or a couple images I should say, that summarizes everything that happened. So if we start on the left, we have meiosis 1. And again, in meiosis 1, the key here is that the homologous chromosomes are separated, whereas in meiosis 2, the sister chromatids are separated. So if we start here on the left, we have prophase 1, where the spindle fibers start to grow. This nuclear membrane will start to break down. Here we have metaphase 1, so those homologous chromosomes, those homologous pairs are going to line up in the middle. The spindle fibers have attached to the chromosomes. Here we have anaphase 1, where the homologous chromosomes are separated. In telophase 1, we have our cleavage furrow that starts to form here, and then our nuclear membrane reforms. Will go through cytokinesis, and at the end of meiosis 1, we have 2 cells. So these are the 2 cells that are going into prophase 2. So you'll notice we still have our sister chromatids paired. So in prophase 2, our nuclear membrane starts to break down, our spindle fibers start to reform. In metaphase 2, M stands for middle, so the chromosomes have aligned in the middle. These spindle fibers have attached to each of the sister chromatids. In anaphase, we separate the sister chromatids. In telophase, we now start to reform that nuclear membrane. We get that cleavage furrow here that is formed, and we'll have a final round of cytokinesis, and we will end up, just like we drew on this previous page, we will end up here with 4 daughter cells that have half the amount of DNA. All right, so the first practice problem that we are going to do is going to be thinking about the number of chromatids that are present. So here we are talking about meiosis 1 and metaphase. And it looks like the formatting got the best of me when I was putting together these pages. And so I'm going to go ahead and fix this really quick. So the answer choices should be A, B, C, D, not all A's. That's kind of impossible. So I apologize for that. Apparently I missed it when I was going through and editing this. But again, bear with me here. So what I like to do for these problems is I always start back at G1. So this is for a normal human. So for humans, we are diploid and 2n equals 46. So at G1, we have 46 chromosomes and 46 chromatids. When we undergo synthesis and we go through G2, so at the end of interphase, we have 46 chromosomes, but each of those chromosomes is duplicated, and so we actually then have 92 chromatids. And so when we go into meiosis 1, we go in with 46 chromosomes, 96 chromatids, and at the end of meiosis 1, we have separated the homologous chromosomes. But we are asking about metaphase. So in metaphase, as this image on the left shows, those chromosomes are lined up in the middle. So the homologous chromosomes pair up and line up in the middle of the cell. And so if we are looking in a microscope, we would see a line of chromosomes, and we would see 46 chromosomes and 92 chromatids. So the next thing that we're going to do, and I promise you there are more practice problems coming here, is we're going to do a summary and a compare-contrast of mitosis and meiosis. So let's go ahead and fill out this table. So for mitosis, we have one division that occurs. In meiosis, we have 2, so we go through meiosis 1 and meiosis 2. For the total number of daughter cells, we have 2 daughter cells at the end of mitosis, and we have 4 daughter cells at the end of meiosis. And in mitosis, the daughter cells are diploid, whereas in meiosis, they are haploid. In terms of role in the animal, so at the beginning of this topic, we talked about why or what are some of the reasons why cells will undergo division, and we said growth, reproduction, and repair. And so now we're going to actually assign these roles to these specific processes. So for mitosis, we use mitosis to replace cells. So if we have damaged cells, we use mitosis for that. For growth of the organism, mitosis is for that. And for meiosis, the entire purpose of it is for making gametes. And again, this is just a little quick short summary of mitosis versus meiosis. And both of these processes of mitosis and meiosis, as you can imagine, are controlled in the cell. So we've kind of drawn chromosomes shuffling around and things moving around, but this entire process is very controlled. And so that leads us to this question of, why do we have control? And specifically today we're going to talk about why do we have control over meiosis and why does it matter? So take a look at the karyotype that we have on the left. So if you look at this karyotype, we have 2 X chromosomes, and so this individual is biologically female. But if you look here at chromosome pair number 21, we actually have 3 chromosomes, and we call this trisomy 21. So tri means 3. So this individual actually has 3 chromosomes instead of 2 for chromosome pair number 21. And so this is more commonly known as Down syndrome. And of course, with Down syndrome, we see a lot of physical growth delays, we see varied intellectual disability, and of course, there are characteristic facial features. So that is a genetic disease that many of us that is more common. Many of us see this, and we may or may not know someone who has this. And we're going to talk about another genetic disease that deals with a malfunction in meiosis, and that is on the right. And this is known as trisomy 18, or also known as Edwards syndrome. So for Edwards syndrome, the majority of The majority of individuals that have Edwards syndrome will actually die before birth, and of those that do survive to birth, most of them, so about 90% of them, will die by the age of 1. And as you can see here in this image, these individuals have a smaller head, they have a misshapen head, they also tend to have smaller jaws, smaller mouths, and noses. They oftentimes are born with a cleft lip and a cleft palate. They also have problems where their intestines can actually grow on the outside of the stomach. They have clenched fists and feet. Many of them also have heart defects and kidney defects. And the reason that we're talking about this goes back to this idea of chromosomal abnormalities and why control over meiosis is important. So let me give you a quick statistic here. In terms of chromosomal abnormalities, it is about 1 in 160 live births. So a little less than 0.5% in there, if I'm doing that math correctly right now. So again, going back to this idea of meiosis, so I've kind of sort of been lying to you, and pretty much anyone who has probably taught you meiosis has been lying to you. And the reason for that is, if we go back to our image that we drew here, we said that at the end of meiosis, we have 4 daughter cells and they're each haploid. Well, it turns out that that's not actually true for everyone. And what I mean by that is, if you've ever heard this idea of, as women get older and as they age, that the chance of having a child with Down syndrome or the chance of having a child with chromosomal abnormalities increases, and you've ever wondered why, it goes back to the lie that I just told you about. So it turns out for meiosis in biological men, when they're producing sperm, it's not a lie. You get 4 sperm that are produced through meiosis. The process is a little bit more complicated than me just saying you get 4 sperm, but at least for biological men, when you go from 1 cell to 4 daughter cells that are haploid, it is true. But for women, that's not actually what happens. We still go through meiosis the exact same way, but it's actually a really cool thing that happens for biological females. When they are developing in utero, all of the eggs or the oocytes that they will be born with have developed already in their ovaries. And this actually happens by the week 12 of development. And as the oocytes for biological females are developing in the ovaries in utero, they actually stop in meiosis 1. And so the entire process of meiosis pauses in meiosis 1 at the beginning at around prophase 1. And then once the biological female actually hits puberty and starts to go through ovulation and gets a menstrual cycle, during the process of ovulation, meiosis actually restarts. And so the female will finish meiosis 1, which at the end of meiosis 1, we get 2 daughter cells. Well, one of those daughter cells is called a polar body. It essentially gets discarded. Nothing happens with that daughter cell. The second daughter cell will eventually go on to become a oocyte through the process of ovulation. When the sperm actually penetrates that egg and fertilization occurs, that is when meiosis 2 happens. So it's kind of mind-blowing that meiosis 2 occurs after the sperm has met the egg. And the reason that we see a higher incidence of these chromosomal abnormalities like trisomy 18 and trisomy 1 as the biological female gets older is because at that point the eggs have been paused in meiosis 1 for decades, and the protein machinery that is responsible for meiosis over time, of course, can degrade. And so the older you get, the more likely it is that something can go wrong in meiosis, and the more likely it is that you can see chromosomal abnormalities in progeny. So although I have kind of lied to you about meiosis with the idea that four daughter cells are produced when I ask you about this in class, we will go underneath the perfect world example, and that is that 4 daughter cells are produced. And if you are more interested in how this process happens in biological males and females, we do actually offer a development course here, and I know that that is covered in that class. So I just kind of wanted to throw that in there as some extra information. So, all right, now that we have fully covered mitosis and meiosis, let's go ahead, and I've got a bunch of practice problems for you all because, like I said, I know that this math is a lot to keep track of and a lot of chromosomes and chromatids to keep track of. So let's start with a practice problem that is all about amount of DNA. So in this practice problem, I am telling you that a cell has 10 picograms of DNA in G2. So a picogram is just a unit of weight. So I'm asking you how much DNA would be present in the following that are listed. So let's highlight the important part. So we have 10 picograms in G2. So if we have 10 picograms in G2, by the time we reach G2, we have duplicated the genome. And so that means at G1, we have half the amount of DNA. And so that would be 5 picograms. So PG is short for picogram. And then the next part is asking us for metaphase, and because it doesn't say metaphase 1 or metaphase 2, you can assume it is asking about mitosis. So during metaphase of mitosis, all of the DNA that we have in G2 goes into mitosis. So we had 10 picograms, so we should have still 10 picograms. At the end of mitosis, we now have 2 daughter cells, and each of those daughter cells should have 5 picograms. So we have gone from 5 picograms in G1 to replication, and in G2 we have 10 picograms to at the end of mitosis, we should have 5 picograms in each daughter cell. All right, so what if this cell, instead of going through mitosis, was actually going through meiosis? So at the end of meiosis 1, which recall, that is where we separate the homologous chromosomes, we would have half the amount of DNA in each cell. So we should have 5 picograms. So we start with 5 picograms in G1. In G2, we have 10 picograms. At the end of meiosis 1, we have 2 daughter cells that each have 5 picograms. Those then go on to go through meiosis 2, and each of those will have 2.5 picograms. So they will have half the amount of DNA that the original daughter cell, which was 5 picograms, had. So again, let me know in the comments below if you get stuck on this question. All right, and then we have a bunch of practice problems, and the ones that we have are asking us about mitosis and meiosis and counting chromosomes and counting chromatids. So what we're going to do is we're going to skip the human ones for now, and we're going to talk about another problem that has pairs of chromosomes, and then I will write in the answers to the human practice problems, but I want you all to have a chance to do this on your own to help you practice for the exam. So let's go ahead and go to page 13 of our notes, and the practice problem that we are going to do deals with an organism that has 3 pairs of chromosomes. So if the organism has 3 pairs of chromosomes, that tells us it likely has 3 from bio mom and 3 from bio dad, which means we have 6 chromosomes total. We see pairs, we should be thinking diploid. So diploid is 2n, and we just said we had 6 chromosomes, so 2n equals 6. In G1, we will have 6 chromosomes. They are not replicated yet, which means we have 6 chromatids. In G2, we have gone through the process of DNA replication. We have 6 chromosomes. which means we have 12 chromatids. And then in metaphase, those chromosomes are lined up. They are not yet separated. So we have 6 chromosomes and 12 chromatids. At the end of mitosis, in each daughter cell, we have 6 chromosomes and 6 chromatids. So again, if you wanted to draw that out, we would start with our 6 chromosomes in G1. In G2, all of those are replicated. So we have 6 chromosomes, but 12 chromatids. And then in metaphase, I'm going to do my best here to draw really small chromosomes. 2, 3, 4, 5, 6. So we have 6 chromosomes that are lined up in the middle and 12 chromatids. And then at the end of mitosis, each of our daughter cells should have 6 chromosomes and 6 chromatids. So now let's do the same practice problem, but instead of mitosis, we're going to be talking about meiosis. So if we have 3 pairs of chromosomes, 2n equals 6, and at the end of meiosis, our haploid cell should have half as many, which means they should have 3. So let's start with G1. G1, we start with 6 chromosomes and 6 chromatids. G2, we have 6 chromosomes and 12 chromatids. So interphase for meiosis and mitosis is exactly the same. And let's go ahead and let's actually draw this really quick. So we've got 1, 2, 3, 4, 5, 6 in G1. And then let's draw G2. All right, now this is going to enter meiosis. And we're first going to I'm going to write M1. So we're first going to go through meiosis 1. And recall that in meiosis 1, we only separate the homologous chromosomes. So at the end of meiosis 1, we will have 2 daughter cells. And in those daughter cells, we will have 3 chromosomes. But each of those chromosomes still has its sister chromatids and is made up of 2 chromatids. So we have 6 chromatids. Now, each of these daughter cells will then go on to go through meiosis 2. And in meiosis 2, that is where the sister chromatids are separated. So we end up with 4 daughter cells that each have 3 chromosomes and 3 chromatids. So the other 2 practice problems that I have in here, and like I said, I will go ahead and fill these tables out, deal with humans. And so thinking about our chromosome numbers and the number of chromosomes and chromatids that we have. And the best way to practice these is to come up with numbers for organisms, for how many chromosomes they have, and kind of work through it that way. So the very last thing that we're going to do that you all probably forgot about is we're going to return to our prokaryotic friends. And again, most of this has been spent talking about eukaryotes, but we got to talk at least a little bit about our prokaryotic friends. So the majority of prokaryotes divide via something called binary fission. Now, there are other ways that bacteria and archaea can divide, but this is by far the most common way. So, unlike humans and unlike many animals that undergo sexual reproduction, and not just animals, but many eukaryotes undergo sexual reproduction, bacteria and archaea do not, and they lead a pretty simple life. So let's go ahead and draw a cell, and here is a chromosome. So they have 1 circular chromosome, and of course that circular chromosome is double-stranded DNA. And just like in eukaryotes where we have to copy our chromosomes, the same thing happens in bacteria. So they are going to copy their chromosomes. So we duplicate the DNA. And then those cells are going to, or I should say that cell is going to get larger the chromosomes will separate. So those chromosomes will separate to the poles. And the way that they separate is using the cytoskeleton. So just like eukaryotes have a cytoskeleton, bacteria and archaea also have a cytoskeleton. And just like we have a cleavage furrow when we get an actin myosin ring, in bacteria at least, we call this the Z-ring. And the Z-ring again is made up of cytoskeleton proteins. And some of them are structurally very similar to those that are found in eukaryotes. They are named different things, but they have very similar functions. And so the idea here, again, is that the cytoskeleton is used to separate the chromosomes to the poles. The cytoskeleton is used to pinch off the plasma membrane to help with the cell wall. And then at the very end of binary fission, we have 2 new daughter cells. And these are identical. And unlike eukaryotes, where the process of mitosis takes 24 hours for quickly dividing cells, for a pretty fast E. coli cell, like a typical average bacteria that's found in your gut, or just in general, it's going to take about 20 minutes. So they are much, much faster at this than we are. But we are actually going to stop there. This wraps up the topic that is all about the cell cycle, mitosis, and meiosis. And we are then going to move into DNA replication. So we have alluded to DNA being replicated both now here on this page, where we have drawn the duplication of DNA in bacteria and archaea. We've also talked about how it happens during the S phase of interphase, so of the cell cycle, and we're actually going to talk about the molecular details of how that happens. So the next topic is all about the history of DNA, and then it's all about how DNA replication actually occurs, and then we will get into how do we go from DNA to protein. So I hope that you all have enjoyed this topic, and I will catch you on the flip side. Have a great day, everyone. Bye.